Good morning. In 2018, Lulu and Nana were two HIV-resistant CRISPR babies produced by Chinese scientist He Jiankui, who claims to have created world's first genetically edited babies. So, uh, He Jiankui has an official YouTube channel, and you can see upon this great scientist, and you can take services of this scientist to produce CRISPR babies and further many more CRISPR services, isn't it? And across the world, many CRISPR uh, therapies and immunotherapies and ophthalmic therapies in different patients is being carried away. Uh, you can see these papers of 12 participants, 14 participants, 72 participants of metabolic conditions, and so on and on. We have also talked about various animal models in pharmaceutical research, and CRISPR in laboratory mouse is also very much famous. Welcome to Murmu Biology, and we were saying Jharkhand eligibility test life science detailed solution question number 70. We come to this question, and this question says knockout mice are generated primarily using the options are RNA interference, diphtheria toxin A chain system, homologous recombination in embryonic stem cell, and chemical mutagenesis. So let's talk in brief what CRISPR is. CRISPR can be understandable in 4 easy steps. First step is you design a guide RNA. This designing of guide RNA can be done with the help of various companies. You design this guide RNA with response to the gene that you are targeting, isn't it? So if you are targeting, let's say, AAAT gene piece, then your guide RNA will be TTTA, isn't it? The second step is, once you have designed the guide RNA, the second step is to produce the Cas9 protein. This also you can buy from biotechnological company, pharmaceutical companies, and the guide RNA is associated with Cas9 protein, isn't it, to form CRISPR Cas9 complex. So this Cas9 complex is then inserted into the target cell, isn't it? So this what this Cas9 complex does is with the help of guide RNA, the guide RNA takes the Cas9 protein to the targeted gene, isn't it? That the AAAT that I have just mentioned, for example, and there the Cas9 makes opens the double-stranded DNA and cuts it, isn't it? So this is CRISPR Cas9 complex, and due to this breaking, the automatic repair system of the cell takes place and the DNA is further repaired, isn't it? So since the gene has been already been cut, so if the gene is whole cut and it is then repaired, then it is known as knockout. And if the gene is replaced from a donor DNA template which can be associated with the Cas9 complex, then it is known as Cas9 knock-in, isn't it? So these are the 4 steps. You can design the guide RNA which will act as GPS, and you will then, the guide RNA is then combined with the Cas9 enzyme, and the Cas9 is inserted into the targeted cell, and the searching of that targeted gene is carried on by the Cas9 enzyme and the guide RNA, and then the targeted gene is cut and then the cell, through its own repair mechanism, it edits the cell. If the targeted DNA is just cut and the DNA is repaired without any other gene, then it is known as knockout. And if there is an extra template DNA carried along with the Cas9 complex, then it is known as new gene insertion is being taken place, and this is known as knock-in. So we come to this question: knockout mice are generated primarily using what techniques? It can be used through various techniques. CRISPR-Cas9 is one of the techniques through which a knockout or knock-in mice can be produced, isn't it? So this is the workflow. The embryonic stem cell is isolated and cultured in a culture plate, isn't it? And the targeted vector, along with the Cas9 system, is inserted into the embryonic cultured cell through various techniques like electroporation or gene gun, isn't it? And that embryonic stem cell is then inserted in the blastula stage or gastrula stage of the pregnant mouse, isn't it? And then when the mouse and the embryo is created, it is either knock out or knock in, and it shows its various phenotype. So this is the workflow of knockout mouse model via homologous recombination. Now let's see upon the what the this is one more image, the embryonic. Let's discuss it once more. Okay, so once the cultured embryonic stem cell is taken out from the pregnant mouse which has been pregnant, the embryonic stem cell is then targeted via CRISPR techniques or via various techniques. The CRISPR-Cas9 system then does it works, and that is knockout or makes the cell knockout or knock in, and then the cells which has been edited is put inside again the pregnant mouse, and then that embryo which is created is now can be called as knockout or knock in mouse, whatsoever is the situation, isn't it? And after that, various combination on various mating techniques are made to obtain and check whether the mouse is knockout or knock in, and so on. So option number A is RNA interference. RNA interference is a biological process. You know about central dogma, isn't it? You just remember, in central dogma the DNA replication takes place, from the DNA the transcription, through transcription the mRNA is made, and then mRNA is helped to make the certain subsequent protein, isn't it? So what RNA interference does, it combines with the mature mRNA and stops the mature mRNA to produce the protein, isn't it? So RNA interference is not related to that gene knockout mouse production. It is rather used to knock down a gene expression, isn't it? So diphtheria A toxin chain system. In diphtheria A toxic A chain system, you must have known the summary of, you must have known, we have just explained how the targeted vector is being used to in the CRISPR-Cas9 to carry in the CRISPR-Cas9 system inside the cell, isn't it? So when we use this vector, we use the diphtheria toxin gene in the last part of the targeted gene, isn't it? Targeted vector gene. So if the vector is associated with the gene, then correctly associated with the gene, then the diphtheria toxin is being cut off, isn't it? Whereas if the diphtheria toxic gene, vector carrying the diphtheria toxic gene, is not correctly inserted into the genome, the diphtheria toxin gene expresses and it kills the cell. So this diphtheria toxic A chain system gene is used for selection purposes, whether the cell has acquired the vector gene correctly or not, isn't it? It is not associated with the gene knockout mice production, isn't it? You can read the whole literature here. The diphtheria toxin chain system is used to it is used for a type of selection system for whether a vector has associated its gene correctly or not. You can read the literature here that just I have mentioned. And homologous recombination is a natural process that takes place during the meiosis, isn't it? You know this homologous recombination, and it also takes place when there is a double stranded break due to any kind of mutation or any certain damage. The double stranded break repair is repaired during using this homologous recombination, isn't it? So this process is used in producing gene knockout mouse. When the CRISPR-Cas9 system cuts the DNA, the homologous recombination takes place to repair the genome, isn't it? So you can see here, I have just mentioned that homologous recombination in meiosis and homologous recombination is used for double stranded break. And chemical mutagenesis is also used for what do you say? Chemical mutagenesis is used for selection purposes, isn't it? Now this is used for expression of specific gene, whether a mutation has taken place, and to further study the mutation for higher purposes, for what kind of mutation has been taken place with that chemical associated chemical, isn't it? So again we will repeat the options. Option number 1 was RNA interference. RNA interference is nothing but a tool for neutralizing the mature mRNA in the process, so that the mature mRNA does not produce any protein, isn't it? Diphtheria toxin gene is used for selection purposes, so that the vector is inserted in the genome or not, isn't it? And homologous recombination, when the CRISPR-Cas9 cuts the genome at the target site, the homologous recombination takes place due to repair the damaged protein, the damage done by the CRISPR-Cas9, and the homologous recombination plays a great part in producing gene knockout mice, knockout or knock-in, whatever may be the situation. This is the literature that you can read upon. Okay. And the chemical mutagenesis is a technique that the chemical is used to produce the chemical the mutation, which is further studied upon, and this has nothing to do with the production of knockout mice. So these are the all the options and the images. RNA interference is not correct. Diphtheria toxin is not correct. It is used for certain certain selection and expression to know whether the vector has taken the correct expression or not. And homologous recombination is correct because after the cutting of Cas9 complex, isn't it, in CRISPR, the homologous recombination takes place to produce the knockout mice. And chemical mutagenesis is the chemicals are being poured upon on the cell to produce different kind of mutation for further study. So you can contribute ₹1 or ₹2 for the fees of the video, and help us grow with us. And talking about CRISPR, you can subscribe, like the video, share and download for offline viewing. Talking about CRISPR-Cas9 in current therapeutic scenario, it is very much expensive, as you can see in the diagram in the image. I will just go through what CRISPR-Cas9 say. Cas9 is there is a therapy known as Cas-JV, which is used for treatment of sickle cell anemia. So you go to the doctor, and the clinical estimation of evaluation of cell collection of your hematopoietic stem cell is around 40 lakhs or 50 lakhs, and then gene editing takes place of that BCR11, if I'm not wrong, BCL11 gene, and that gene editing takes gene editing and designing of guide RNA takes place around 9 to 12 crores, isn't it? And monolab activation, your whole what do you say, the whole hematopoietic stem cell present in your bone marrow is flushed out. It takes another 2 crores, and then the new CRISPR-Cas9 edited cells that have been cultured and multiplied is being new cells is being new hematopoietic stem cell is infused into your bone marrow. This again takes place another 1 crore, and then your recovery and long-term monitoring consists of 5 crore, and the total estimates is around 20 to 25 crores. So this is just a not practical for the situation. And thank you, stay tuned for